Good evening, everyone, and welcome to the monthly Otto Leopold Audubon Society meeting. I hope this evening found you all well and happy, and as we hopefully move into the waning days of COVID-19 isolation. Tonight's program was originally scheduled for last spring, but due to the pandemic was moved to now. The timing of the program was not by chance. We all need this time of year a little chance to get into a sunny, warmer climate. So tonight, we're going to explore the underwater world of the Coral Triangle, a body of the ocean north of Australia that covers an area roughly the size of the western United States. The area is one of the most biologically diverse places in the world, a marine Amazon forest that contains 75% of the world's coral species and 37% of the reef fishes. Our guide for this evening's journey will be Audubon's own inimitable Karen Dostal, longtime board member and teacher in the Stevens Point school system and former director of the Boston School Forest. As a matter of fact, both of our children had Karen as their fourth grade teacher. Karen is active in all sorts of outdoor activities. She devotes time to yoga, gardening, hiking, biking, swimming, cross-country skiing, and birding, and also scuba diving. So in 2017, Karen and her husband, Mark, spent a week diving in a small portion of the Coral Triangle, bringing back some incredible video footage of the diversity of marine life found in the area. She'll also tonight describe the threats to that region and what's being done to try to protect and conserve the area. So, without further ado, let me turn this evening's program over to our esteemed speaker, Karen Dostal. Karen?
So the story of why we went to this region of the world on a once-in-a-lifetime trip, really, is sort of embedded in the program. And when I made this, I started with sort of a PowerPoint slideshow and then converted it into an MP4 file, took it into iMovie, and embedded other video into that. So basically, I'm going to play this. It's going to run for about 48 minutes. And then after that, we'll have a short question and answer session. I think this evening, you'll see things that, animals that you fish, other creatures you've never seen before. And I introduce some of those, as many as I could, to you before each of the video clips. When Mark and I got back from the Indonesian trip, I took a bunch of the video clips of the underwater scenes that we had, and I put them into two 15-minute videos called Diving in the Heart of the Coral Triangle, parts 1 and 2. And I put those on my YouTube channel, and then also embedded them into this program tonight. And turn your sound up. There's really beautiful music in the background. So just relax and enjoy, and we'll talk more at the end of the program.
During this program, I will cover where the Coral Triangle is and the unique social and political aspects of the region.
Karen, it's not sharing yet.
Change on corals will be discussed, as well as how the Nature Conservancy work to protect the Coral Triangle. Before we go under the sea, you'll...
Again, we did hear the sound, but we didn't see the screen, so.
Okay. So I'm going back to share screen, Susan?
Yes, that's correct.
Okay.
There you go. Perfect. So we see your screen now. Oh, it's doing that, isn't it? Well, we see your screen.
Okay, well,
Yep.
let's see if it'll play now.
Yep.
Now-
Coming through great. Yep.
During this program, I will cover where the Coral Triangle is and the unique social and political aspects of the region. Corals and the impact of climate change on corals will be discussed, as well as how the Nature Conservancy work to protect the Coral Triangle. Before we go under the sea, you will learn a bit about scuba equipment, especially how it allows us to breathe underwater. Then I will play the videos, Diving in the Heart of the Coral Triangle, Parts 1 and 2. The Coral Triangle is in the Western Pacific and lies between the South China Sea, the Philippine Sea, and the Timor Sea to the south. Countries it borders or surrounds are the Philippines, Indonesia, Papua New Guinea, Malaysia, the Solomon Islands, and Timor-Leste. The Coral Triangle is known as the most diverse marine ecosystem in the world, containing 30% of all coral reefs and 75% of all coral species and 37% of the world's reef species. Like most coral reef systems around the world, the Coral Triangle is under threat from habitat change, rising demand, intensive harvesting, and climate change. Over 120 million people live in the Coral Triangle and depend on its coral reefs for food, income, and protection from storms. And so, the need to protect and preserve this biome became very apparent. We will learn more about how this was accomplished in a bit, but first, I'd like to explain more about what coral is and how human-induced climate change is affecting the reefs around the world. In this clip, Mark is chasing a black and white spotted barramundi cod. But try to take a close-up look at the corals. Most structures that we call coral are in fact made of hundreds or thousands of tiny coral creatures called polyps. Each soft-bodied polyp, most no thicker than a nickel, secretes a hard outer skeleton of limestone or calcium carbonate that attaches either to rock or the dead skeletons of other polyps. In the case of stony or hard corals, these polyp conglomerates grow, die, and endlessly repeat the cycle over time, slowly laying the limestone foundation for coral reefs and giving shape to the familiar corals that reside there. Because of this cycle of growth, death, and regeneration among individual polyps, many coral colonies can live for a very long time, some 25 million years old. Most corals live in symbiosis with algae called zooxanthellae, which are plant-like organisms. It is the zooxanthellae that give corals the beautiful colors we see. Residing within the coral's tissues, these microscopic algae are well protected and make use of the coral's metabolic waste products for photosynthesis, the process by which plants make their own food. The coral polyps benefit in turn as the algae produce oxygen, remove wastes, and supply the organic products of photosynthesis that corals need to grow, thrive, and build up the reef. Climate change is causing our oceans to warm. Remember the algae that resides in symbiosis with the coral polyps? The warmer water causes coral polyps to expel the algae, causing the coral to bleach. The bleaching doesn't kill the coral polyps initially, but it stresses the corals. The corals can recover when conditions change. Sedimentation, more frequent storms, altered ocean currents all add to stress on the reef systems. Remember also, the coral polyp's hard outer skeleton is calcium carbonate. As the oceans absorb excess CO2 due to the burning of fossil fuels, the oceans are becoming more acidic. Acidification dissolves the calcium carbonate structure of the corals.
Karen, did you push pause on the presentation?
No.
Our friend and
Hey.
UWSP
Is it
alumni,
working?
Sheldon Cohen, works for the
It is.
Nature
Yes.
Conservancy in Washington, D.C. He and his family, Tricia Hansen, who is also a UWSP alumni, and their son Zach, moved to Bali as Sheldon was about to head up the Coral Triangle Initiative from 2006 to 2009. The Nature Conservancy organized a process involving the 6 Coral Triangle countries through a set of senior officials meetings to negotiate an ambitious regional action plan for the Coral Triangle. The action plan was formally adopted by the heads of government at their summit in May of 2009. The 6 countries set up a permanent secretariat in Indonesia to help support implementation of the Coral Triangle Initiative. The process has continued the past 11 years, involving annual minister meetings and wide-ranging collaborative action and country-specific action on such topics as marine protected areas, sustainable fisheries, threatened species, nature-based adaptation to climate change, and women-led coalitions. One other outcome of the Coral Triangle Initiative is the development of the Coral Triangle Center on the island of Bali in the town of Sanur. Here's a photo of Sheldon with one of his colleagues, Rili Johani, from their days working to establish the intergovernmental agreement that became the Coral Triangle Initiative. The Coral Triangle Center, with Rili at the helm, promotes conservation of marine biodiversity and sustainable management of marine and coastal resources across the Coral Triangle. Although Sheldon worked for several years on the Coral Triangle Initiative, he had never actually been to the most marine diverse region of the Coral Triangle. So, in 2016, we began planning a trip to dive in the Raja Ampat region of the Coral Triangle. On Friday, April 13th, we flew from Chicago, and Sheldon and Trish from DC, to Doha, Qatar, and then on to Bali, Indonesia. We explored Bali by riding bikes down Mount Batur, whitewater rafted the Angong River, and found the people, food, and culture absolutely amazing. After a week on Bali, we flew another 7,000 kilometers to Sarang, West Papua. It was then a 2.5-hour boat ride from Sarang to the island of Cree. Note the group of islands circled to the west. These are the Fam Islands and will come up a bit later in the program. Here's the island of Cree. Note the marker on the east end of Cree called Cape Cree. It is here that a world record of 374 fish species was documented in 2012. You will see an example of this in the following videos coming up, right after you learn about dive equipment and how we breathe underwater. First, a peek at Cree Eco Resort. On the left, our first glimpse of home for the week from the boat and the Papuan-style cottages we stayed in.
Okay. Hi, I'm Bob from Dive Point Scuba in Stevens Point, and giving you a little rundown on how a scuba regulator works. To start with, I have a tank here, a scuba cylinder, and the scuba tank holds air. All right, we generally use air breathing. There are other mixes. This is marked for nitrox, which is a higher percent of oxygen. But normally we just breathe air, and the air in the tank is just like atmospheric air we're breathing right now. It's under high pressure, so we can get a lot in there. This tank right here holds about 80 cubic feet of air, and a cubic foot is about the size of a milk crate. So the air in 80 milk crates is compressed at high pressure into the tank, and the pressure that it has to get to get that amount of air in there is about 3,000 pounds of pressure. A bike tire has 30 to 60 pounds, so it's about 100 times more pressure than that to hold that amount of air into the tank. All right. To get the air out of the tank, we have a regulator system. All right, and the regulator system consists of two stages. The first stage attaches directly to the valve on the tank, and the first stage uses a valving mechanism that takes tank pressure that's at 3,000 pounds when it's filled and breaks it down with a valve system in there. It can be rather complex. Breaks that pressure down to about 150 pounds of pressure, and that is about the pressure that you get out of an air compressor used in a garage or power tools and things like that to fill your bike tire. So inside here, the valving mechanism takes it 3,000 through the valving mechanism down to about 150 pounds of pressure, and so the air in the hose here is actually only at about 150 psi. All right, that pressure is dependent also on depth. As we go deeper, all right, the regulator senses the surrounding pressure change because you're going under more and more pressure, and the pressure going to the second stage actually increases and goes up. If it would stay constant, at a certain point we would not be able to get air out of the regulator because our surrounding pressure would be greater than that 150 pounds of pressure. All right, so it's also 150 psi, pounds per square inch, over our ambient or our surrounding pressure. When it gets to the second stage basically has, all right, a diaphragm, and when we inhale, all right, we create a vacuum inside the regulator, and that vacuum pulls a diaphragm in. That diaphragm, I'm going to pull it out here, that diaphragm then pushes on a lever mechanism, all right, and that lever mechanism is called a downstream valve. So the pressure in the hose coming into the second stage pushes against the low pressure seat, what we call it, and as we inhale, that lever opens and the seat opens, allowing the air to flow into our mouth. So it's dependent on the amount of vacuum that we generate when we inhale. So it is a kind of a mechanical breathing. When we stop breathing, the valve will close and the flow of air stops. And then when we exhale, there is a one-way exhaust valve, like a flutter valve on the bottom of a snorkel, in the bottom of the regulator. And when we exhale, that air then goes out through the exhaust valve. What I'm going to do is turn the flow of air on. When I turn the air on, again, the air comes through, nothing happens, all right, until we movement on the lever, and that allows the air to flow into your mouth. When I put the diaphragm back on, you can actually see what happens. When I inhale, I'll kind of hold the diaphragm in place. When I inhale, it pulls the diaphragm in, and that again pushes on the lever and allows me to inhale. And some of the regulators have adjustments on them for flow and things like that to make it easier to breathe as you get deeper and things like that. This is a commonly referred to as octopus regulator, an extra second stage. When we dive, we always have a primary regulator, the one we're going to breathe off of, and then some way for a partner to breathe off of. So we always dive with a buddy. So if my buddy would run out, I have another second stage I can pass off to him, or another type of that system would be a built-in regulator onto our vest, our buoyancy control vest. So if my buddy would run out of air, low on air, I can pass the regulator I'm breathing off of, and I can breathe off of one that is hooked to the rest of my unit. As you run down in pressure, all right, you get to the point where if you're running out of air, again, it gets harder and harder to breathe. But before you do that, you're usually monitoring your gauges so you can monitor how much air you have in your tank. And I don't have a gauge hooked up to here, but just basically a gauge telling you how much air is in the tank. And we never try to run out. We always want to save some reserve, so we do have enough air so that we can get out of the water safely. Anything else?
Clockwise, let me introduce you to Teo, Albert, and Dowd, our boat crew for the week. Crete Eco Resort was the first dive resort in the Raja Ampat area And rather than bring in dive personnel from the outside, they trained local islanders. By employing local people, Cree Eco Resort is helping to improve the lives of local inhabitants, and this was very evident when walking through their nearby village. Usually dive resorts have PADI certified dive masters to lead the dives. Teo didn't have that certification, but he knew the reef well and did a fantastic job. In a further departure from a typical dive center, there were no life jackets on board the boat, no emergency kit or oxygen in case a diver got decompression sickness. So our maximum depth was always 20 meters, or around 60 feet. Language was a barrier on land, but underwater, we communicated perfectly well with common divers' hand signals. As we rolled backward off the boat on day one, we entered a wholly new underwater world from what we knew of the Caribbean. Many fish, corals, and mollusks were familiar, like angelfish, damselfish, and parrotfish, were recognizable, but the shape, coloring, and size differed. Then there were the creatures endemic to the South Pacific, like this black and white Moorish idol. In this first video, you'll see more of these fish. They're called sweet lips. You'll also see Titan triggerfish, hawksbill turtles, pipefish, Napoleon wrasse, yellow-tailed barracuda, sea cucumbers, giant clams, lionfish, white-tipped sharks, clown triggerfish, anemones and their associated clownfish, moray eels getting their teeth cleaned by wrasse, leaf scorpion fish, bumphead parrotfish, two different woebegone sharks, and especially a lavender-colored pygmy seahorse on a lavender sea fan coral.
That's the thing they have on their t-shirt. It's only the first one, right?
At the end of our week, the whole crew from Crete Eco Resort took us and the rest of the divers there on an all-day excursion to 3 new dive sites. The first was Manta Sandy, a dive site where the giant manta rays come in to a cleaning station. The cleaning station is a coral head where fish gather. The mantas come in, and the fish eat parasites on the manta's skin and in their gills. The second site was called the Passage. This was a shallow dive between a maze of islands. There was little coral here, but there were many creatures to see because they had little cover. You'll see seahorses, immature cuttlefish, blennies, and colorful worm-like mollusks called nudibranchs. The third dive was off the end of the island where the arrow points in the photo on the left. At the end of part 2 of the videos, you'll see swarms of colorful fish, but look in the background for healthy, beautiful coral as far as the eye can see. It was breathtaking. Look out for the banded sea snakes, they're very venomous. And wait for the giant spotted octopus. Notice how it can change the color and texture of its skin in an instant. Octopus are masters of disguise due to cells in their skin called chromatophores. Chromatophores are attached to a nerve ending, meaning the expansion or contraction of the chromatophores is controlled by the nervous system. When a predator or prey is detected, the octopus brain sends a signal to the chromatophores. Some scientists speculate that the octopus skin is, in a sense, an extension of the brain. You will see the octopus spread its mantle when it has caught something to eat.
That's it.
Wow, that was awesome. So everyone in the chat, go ahead and put your favorite part. Go ahead, tell us what your favorite part is. If you have questions, you can use the reactions button and raise your hand and I can call on you, or you can unmute yourself and ask a question, or you can use the chat to ask Karen some questions. We've got the octopus. Coral reefs were so beautiful. It was incredible. Wow, amazing. Amazing diversity. Thank you, Karen.
Yeah.
Yeah, it was incredible. That octopus at the end. Have you seen the movie or the documentary? My
Octopus teacher.
Yes.
Yeah. We spent a full 10 minutes with that octopus underwater. And,
Wow.
you know, I only put like less than 2 minutes in the video, but yeah, we watched it feed 3 times. It was not concerned that we were there at all.
So cool. How much experience do you have with diving to do something like this? How much experience do you think you'd have to have for something like that?
Oh, how much you'd have to have?
Mm-hmm.
I'd say you'd have to have your open water certification and go with an experienced diver. And I don't know. So, I mean, the biggest challenge there diving were the currents. At the end of the first video, you can see how we're just flying along at a jogging pace. And so you'd have to be prepared for that.
Okay.
Are you ready?
Sue, did you have a question? Kent Hall?
Yes, I have a question. First of all, what a wonderful series of films you did, Karen. It's just terrific. I've done quite a bit of diving myself, and I've never seen so many nudibranchs as I saw in your shots. They're beautiful.
Yeah, they were really something. It was fun to see.
Yeah,
Anyone else?
How much experience do you have, Karen? I mean, have you been diving, you know, your whole life or?
Yeah, about 10 years. I have about 300 dives in.
Wow.
Mm-hmm.
Oh, that's great. There's lots of comments. You can read those. How much diving time did you get when you were there? Someone asked.
We did 3 or 4 dives a day. So we'd be down, 15 minutes, maybe an hour if we were lucky. And then some of the video where it looked very dark blue, that's where we were down deeper, especially at sunset. And that's where we were seeing the sharks and the giant trevally and just all those scads of fish off the end of Cape Cree there, where they documented the highest number of fish species ever counted in a spot. So.
Karen, I had a question. When we were looking near the end at the octopus,
Yep.
and you talked about the chromatophore distribution of pigment earlier, I'd never seen quite what you saw there. I've done some scuba diving myself in the tropics and coral reefs, but at one point it looked like it was all this blue color, and I'd never seen that over a significant portion of the far edge of the arms of the octopus.
Oh yeah, that's where it was spreading out its mantle and it caught a fish, or it caught something to eat. Yeah, and it did look quite blue. Some of that is just the depth, and the deeper you go, you lose your red in the spectrum first, gets filtered out by the depth of the water, and you end up with mostly blues. That's why when Mark has the GoPro going, he's got different red shields that he puts on at different depths to color correct. And then I did take the videos in and I put them through some color correcting software so they look a little more normal, like what you'd see under the water.
And my other question was about the banded sea snakes. You pointed out that they're dangerous.
Yeah.
It looked like they were hovering right along the bottom.
Yeah.
Are they dangerous if you just stay back about 10 feet or so, or
they're not
attack humans?
I mean, if you see most of what you see down there, it's really not interested in you as a diver. The sharks are going by, they're not looking for people. They're mostly dangerous for people who live in the area, and they come out of the water at night. They'll fill up, their lung is almost as long as their body, and they fill up with air, and they can go down for hours under the water. And then they come out at night, and you just wouldn't throw your sleeping bag down on the shoreline probably if you knew they were around.
Marvelous photography. I just thoroughly enjoyed it. Just amazing.
Yeah, it's fun to do.
Yes, there was another question on, did the spotting the sharks make you nervous? You guys did get pretty close to that.
Yeah. No, not those sharks.
Oh. And were they white-tipped sharks or something? It looked like they were.
They were white-tipped sharks, yeah. The other sharks I want to point out were in the first video, they're super camouflaged, and it looks like they have all this algae growing off. Those are called woebegone sharks, like Lake Wobegon.
Oh, wow.
Because they look so pathetic. I guess, I don't know.
That's funny.
That's actually a shark, yeah. So.
There was a question about recording, and yes, this program has been recorded. We post all of our programs on the YouTube channel, the Aldo Leopold Audubon programs. We also have a new website coming up. It should be live by the end of the month, so stay tuned for that. So we're just making some revisions for that, and our YouTube channel will be linked to that new website as well, so to keep you all informed. Other questions from anyone? I'm kind of going back and forth here, trying to monitor the chat, see if someone raised their hands. Anything else?
No.
I will just make a mention of our next program coming up then. And next month, we do have the Kirkland Warblers in Wisconsin on February 17th. That one is available. You can register for that as well. Again, it's free and open to the public. We have a biologist from the Wisconsin DNR giving us an update on that species and sharing a lot of the research and data on it. So it's stay tuned for that.